The polymerase chain reaction, or PCR, was invented by the American scientist Kary Mullis in the early 1980s. PCR allows for amplification of specific DNA segments from purified DNA preparations as well as environmental, clinical, or forensic samples. Today, PCR is a fundamental tool in biology, biotechnology, medicine, and forensics. There are two main ways in which PCR can be used. Analytical PCR is used to detect, characterize, and/or quantify a particular DNA segment. For example, is the specific DNA segment present within the sample or not? What is the length of the amplification product? Is there more than one amplification product? PCR can also be used to measure how much of a particular DNA segment is present within a sample. This is known as Quantitative Pcr, or simply qPCR. Preparative PCR is used to amplify a DNA segment either in order to clone it or to sequence it. A PCR reaction mixture must contain the following components: a DNA template, a pair of single-stranded DNA oligonucleotides, typically 18 to 30 nucleotides long, to act as primers, a thermostable DNA polymerase, an equimolar mixture of all 4 deoxynucleoside triphosphates. Finally, buffering agents and salts, especially magnesium, are included to create the appropriate chemical environment for the DNA polymerase to function. Tac Polymerase is the most commonly used thermostable DNA polymerase for PCR. Tac Polymerase is derived from the hyperthermophilic bacterium Thermus aquaticus, which was originally isolated from a geothermal spring. Once all PCR reagents have been combined, the PCR reaction can be initiated and sustained without having to add any further reagents. All that is required in order for amplification to occur successfully is to incubate the reaction mixture at a specific set of temperatures and time intervals for a number of cycles. Thermocyclers, commonly referred to as PCR machines, are used for automated control of incubation temperatures, time intervals, and the number of cycles of the PCR reaction. Before looking at the PCR reaction, it is necessary to first take a closer look at how DNA itself is replicated. The first thing we must be aware of is that nucleic acids such as DNA have polarity, that is to say, they have directionality. This means that one end of a single DNA strand is chemically different from its other end. Let's start by looking at the structure of deoxyribose, the sugar component of DNA. Deoxyribose is a 5-carbon sugar. The convention is to number the 5 carbon atoms 1' through 5' as shown. The purine or pyrimidine nucleobase will attach to the 1' carbon. The 3' and 5' carbons will connect to phosphate groups to form phosphodiester bonds that links to neighboring deoxyribose molecules in the DNA backbone. Here is a schematic representation of a short double-stranded DNA molecule. Note that the two strands run in opposite directions to each other. To put it another way, the two strands are anti-parallel. We now highlight the terminal carbon atoms in the backbone of each DNA strand. This gives rise to the convention of calling one end of a DNA strand the 5' terminus or the 5' end, while the other is referred to as the 3' terminus or the 3' end. Note that the 5' terminus of one strand will be opposite the 3' terminus of the other strand. DNA is synthesized from a free 3' terminus in the direction 5' to 3' from the perspective of the strand that is being synthesized. In this particular diagram, this corresponds to the left strand. DNA synthesis also requires a template strand in order to ensure that the correct nucleotide base is added to the growing DNA molecule. This is another schematic representation of the DNA synthesis reaction, with each base indicated by a letter. 5' and 3' termini are indicated at the ends of each strand. A primer sequence, shown here in red, provides the free 3' terminus from which DNA synthesis can be initiated. The template strand, which is anti-parallel to the primer sequence, is shown in blue. A DNA polymerase initiates DNA synthesis from the 3' terminus of the primer and proceeds in the direction 5' to 3' from the perspective of the strand that is being synthesized, which in this diagram is the red strand. According to standard convention, the upper or top strand, shown in red, is always shown with a 5' terminus to the left and the 3' terminus to the right. The termini are of course reversed in the complementary strand, shown in blue. Now we can take a more detailed look at the actual PCR reaction itself. For this example, we will use a hypothetical template DNA shown here as 2 horizontal lines. The 2' sites are indicated in red and blue. In this example, the template appears to be a relatively short linear DNA molecule. However, in reality, the template DNA can be millions of base pairs long. It can also be a circular rather than a linear DNA molecule. In addition, the DNA template can consist of a complex mixture of thousands or even millions of different DNA molecules. For clarity, we will use the current hypothetical template. Another thing to keep in mind is that the reaction mixture contains a significant excess of primer molecules relative to the DNA template molecule. At the start of the reaction, primer molecules will generally outnumber the template molecules by a factor of 1 billion or more. The first cycle of the entire PCR reaction is initiated by heating the reaction mixture to 95 degrees for between 30 seconds and 1 minute. This causes the 2 DNA strands of the template to separate, a process known as denaturation or melting. The temperature is then lowered to somewhere between 50 and 65 degrees for 30 seconds to 1 minute. This allows the 2 primers to bind to the primer sites of their corresponding template strands. This process is known as annealing or hybridization. Note that the 3' termini of the primers point towards each other and the center of the DNA sequence to be amplified. Also note that the primer sequence is complementary to the primer site on the template strand. Conversely, the primer sequence is identical to the primer site of the opposite strand. The specific annealing temperature depends on the length of the primer as well as the proportions of C and G nucleotides to A and T nucleotides. Longer primers require higher annealing temperatures. Likewise, primers with a higher proportion of C or G nucleotides will require a higher annealing temperature. If the annealing temperature is too high, the primers will not bind efficiently to the template. If the annealing temperature is too low, there is a risk of non-specific binding of the primer to the template, which can lead to amplification artifacts. The temperature is now raised to 72 degrees for anywhere between 30 seconds and 4 minutes. 72 degrees is the optimum temperature for the Tac polymerase. Tac polymerase initiates DNA synthesis from the 3' terminus of each primer and proceeds to synthesize a new strand of DNA in the direction 5' to 3' from the perspective of the strand that is being synthesized. Note that the DNA synthesis does not necessarily stop at the end of the opposite primer site as long as there is a template strand and the temperature continues to remain at 72 degrees. The duration of the elongation step depends on the length of the sequence to be amplified. Tac polymerase typically requires 1 minute for the synthesis of 1,000 base pairs. Under standard conditions, Tac polymerase can synthesize up to 5,000 base pairs before dissociating from the template strand. The cycle is now repeated for a second time. Note that at the end of the second cycle, DNA molecules have now been produced that have blunt ends on one side. The cycle is now repeated for a third time. At the end of the third cycle, DNA molecules have now been produced that have blunt ends on both sides. These products will become the dominant species during the PCR reaction, as will be explained in a moment. The PCR reaction will continue in the same manner for up to 30 to 40 cycles. Let's take a look at the diversity of DNA products at the end of cycle 3 and how their proportions will change as the PCR reaction proceeds. If we run the reaction one more cycle, the proportions of the different products will look like this at the end of cycle 4. There are essentially 3 kinds of DNA products formed during the PCR reaction. The first kind of product possesses single-stranded overhangs on both ends. These products derive directly from the original template DNA and will therefore not increase in number as the PCR reaction proceeds. The second kind of product possesses a single-stranded overhang on one end. The number of these products will be amplified in a linear fashion as the PCR reaction proceeds. The last kind of product possesses no overhangs, that is to say, both ends are blunt. The number of these products will be amplified in an exponential fashion as the PCR reaction proceeds. They will therefore quickly become the dominant DNA product in the reaction. Provided that the amplification process is 100% effective, the DNA sequence between the 2 primer sites will become amplified by a factor of 10 trillion times after 40 cycles.